Hello everyone, welcome to Mediverse Atlas. In today's video, we are going to talk about cell-mediated immunity. As the name suggests, cell-mediated immunity. That is, immunity is mediated by some cells. Now, what are these cells? These are T lymphocytes. So, obviously, cell-mediated immunity is the immune response mediated by T lymphocytes, and antibodies are not involved in cell-mediated immunity. Why antibodies are not involved? Because T lymphocytes are involved. So, there is no need of antibodies to generate an immune response. Next, cell-mediated immunity, it provides defense against intracellular pathogens such as viruses, fungi, certain bacteria and tumor cells. Okay, and as discussed in the earlier video, T lymphocytes are of four types: cytotoxic T cells, helper T cells, suppressor cells and memory cells. Now, let us dive into the steps of cell-mediated immunity. In total, there are totally 3 steps of cell-mediated immunity. We'll discuss every step in detail. The first step, antigen recognition, processing and presentation. So, antigen must be first recognized, processed and it must be presented to T lymphocytes. And how does this process take place? We'll be studying. First, we will be talking about processing and presentation of antigen. So antigen itself is not recognized by T lymphocytes. So it must be get processed and it must be presented to the T lymphocyte for the generation of immune response. So how does this processing and presentation takes place? First, when there is an antigen encounter in our body, there is phagocytosis of antigen by antigen presenting cells. Okay, by which cells? Antigen presenting cells. There will be processing the phagocytosis of this antigen. After the phagocytosis, there is partial digestion of antigen into small peptide fragments inside the antigen presenting cell. And in the antigen presenting cell, there will be synthesis of MHC molecule, that is the major histocompatibility molecule. We have discussed MHCs in detail in the previous video. If you have not watched, I'll provide link in the description. Please go and check it out. So synthesis of MHC molecule takes place in the antigen presenting cells. Then after the synthesis, there is fusion of antigen fragment with this MHC molecule. So after the fusion of antigen fragment and MHC molecule, there will be exocytosis of this complex, the antigen and the MHC complex onto the surface of antigen presenting cells. So this is the process of presentation of antigen to T lymphocytes. I have represented this in a diagrammatic format here. So first, this is the antigen. There will be phagocytosis of antigen by antigen presenting cell. After the phagocytosis, there will be partial digestion into peptide fragments. After it, there will be synthesis of MHC molecule. After synthesis, there will be fusion of this MHC molecule with antigen. After fusion, there will be exocytosis, that is the presentation of antigen with the help of MHC molecule onto the surface of antigen presenting cell. This is the process of processing and presentation of antigen. Now we'll be studying about recognition, like how does T cell recognize that the antigen is presented. Next, we'll talk about recognition of T lymphocytes. Recognition of antigens by T lymphocytes. So recognition of antigen by T lymphocytes. Antigen is recognized by T lymphocytes with the help of T receptors on T cells. So on this, see, this is the T cell. On the surface of T cell, there are some receptors known as TCRs, that is T cell receptors. These TCRs help in the recognition of this the MHC antigen complex, which is present on surface of antigen presenting cells. Okay, these TCRs bind to the antigenic peptides that are presented on MHC molecule on the surface of antigen presenting cells. See, in this diagram, see, this is the antigen presenting cells in which processing takes place. Then it is presented on the surface of antigen presenting cells. After that, T cells, they recognize with the help of TCR, which are presented on the surface, and they go and bind to the antigen fragment of the this complex, MHC AG complex. Okay, I hope you got this. You got this concept. Next, the second step, the activation and proliferation of T cells. Now after the binding of T cells with this antigen, these T cells get activated and get proliferated to generate an immune response. Now, how does this process take place? We'll be studying. Next, T cells, both T4 and T8, that is T helper and T cytotoxic. These both cells get activated as soon as TCRs come in contact with MHC antigen complex. Next, activation of T cells is the first step in induction of CMI, that is cell mediated immunity. So the first induction step in the cell mediated immunity is the activation of T cells. Now, when does this T cells activated? As soon as they get attached to this complex. Okay, I hope you got this point. Now here, how does the proliferation also takes place? Now see, we'll be studying this flowchart. Antigen is presenting on MHC2 AG on its surface. Obviously, if MHC class 2 is there, which T cell come and binds? T4 cell come and binds. We have discussed in the earlier video 24 for T helper cells, right? T4 cells. MHC, if the antigen presented is with the complex of MHC2, T4 cells come and contact. After that, they get activated and they get proliferated. After the proliferation, these cells are divided into two types. One is the memory cells and one is the effector cells. Okay, T memory cells and T effector cells. That is, and in T helper cells also we have types, right? T helper 1 cell and T helper 2 cell. In this case, T helper 1 cell, they releases interleukins. After the release of interleukins, there will be co-stimulation process. These two processes takes place simultaneously. Co-stimulation of what? We'll be studying this now. Next, now virus infected cells or the antigen presenting cells with MHC1. If it's MHC1, we have discussed 18. That is the cytotoxic T cell, like this TC cell. Okay. Next, T8 cells or the TC cells, they come and bind and get activated. After the activation, they get differentiated and proliferated. In this differentiation and proliferation, this co-stimulation with the help of interleukins 2 takes place. Okay, effector T4 cells, they release interleukins which help in co-stimulation of proliferation and differentiation of T8 cells. After this, T8 cells get divided into effector cells and memory cells. They synthesize effector cells and memory cells. And these effector cells, which effector cells? T8 effector cells, they kill the invader, I mean the microorganism or the pathogen, mainly by which process? Cytolysis, that is lysis of cell. Okay, breakdown of cell. Okay, breakdown of what cell? The cell of a pathogen. Okay, I hope you got my point. First, there is T4 cell, T8 cell are, I'll just write in a brief format. T4 cell is activated, T8 cell is activated. After that, T4 cells, that is T helper 1 cell. In T4 cells we have T helper 1 and T helper 2. So it is T helper 1 cell, they release interleukins 2 and co-stimulates the proliferation of T8 cells. T8 cells are proliferated into the effector cells and the memory cells. These effector cells, they kill the pathogen. Okay, this is the main process. Now the last step in the cell mediated immunity is the elimination of invader. Next, cytotoxic T cells, that is T8 cells, mainly eliminate the invader by three mechanisms. One is the cytolysis, secretion of lymphotoxin and interferons. So this is the actual concept of cell mediated immunity. You can represent in this manner only in the exams to score good marks. And please don't forget to follow our Instagram and as well as Telegram. Links are in the description. And thank you for watching this.